It's a real pleasure of mine to be able to introduce Soren to the team. For this webinar series. Soren has been part of our team for many, many years now. And I see some of his colleagues in Denmark and during the call too. So this is great. I just wanted to say that, Soren sort of entered our group as a PhD student at Aarhus University in Denmark, where he was doing a lot of work on species mapping of forage mixtures using computer vision and AI applications. And so now he's sort of deeply involved in his postdoc capacity in PSA, working with us all around the development of the weed image repository, working to do the real-time weed quantification using the OAK-D systems. And this is all part of the pipeline of tools that are entering both the PSA network as well as the GROW network. And Soren has been sort of a key leader in all of this space. And so we've just been real privileged to work with him. For those who don't know, a big part of the image repository work that we're doing in the US is really being modeled after what the Danes did. And Soren was sort of the workhorse in that effort. And so he's really bringing a tremendous amount of expertise to our team. And we are just deeply in debt to all of both the wisdom that he has to offer and the great camaraderie that he's brought to our team. So with that, I welcome Soren to present on his work.
Thank you so much for this presentation. Oh, sorry, for this introduction. Let me just share my screen.
No, okay.
Do you hear me all right? Great. And if you have any questions during the talk, just interrupt. I'm happy to sort of dive into any subject that you might want to hear more about. So as Stephen said, this is a short presentation on field scale mapping of crops and weeds using high resolution cameras and deep learning for computer vision. So the reason we want to do this, well, precision ag is all about doing something on the field, not based on what we think is going on, but what is actually going on. So this, in my work, this involves taking images. So going out to the field, collect images in one way or the other, do some machine learning, computer vision to actually digest that data into something that we can use. Then act on that data. So that could be mixed crop composition, it could be fertilization maps, it could be deficiency maps, it could be spray maps for herbicides, it could be pretty much, well, it could be a wide range of maps. And then we could apply that in the field where it's sort of reacting specifically to what's going on in that spot in the field. The start of this presentation takes off in my PhD. So basically, I've been working on mixtures of grasses and clovers. So what you see here is the left is ryegrass, the right is white clover. For the dairy industry, we want to see both as a mixed crop to maximize the value of the crop. But depending on a multitude of factors, so it could be the weather, precipitation, the temperature, soil composition, then we get a different sort of species composition in the field. So in the left side, you see very low legume parts. In the right side, you see quite a legume-rich stand. So since legume is a nitrogen-fixing crop, we want to apply the fertilizer dependent on the legume content here. So in the left-hand side, we want to apply as much fertilizer as possible to maximize the yield. In the right-hand side, we want to minimize the fertilizer amounts so we can maximize the legume content and then maximize the protein content and the value in terms of dairy production. Yes. So the idea behind this project is basically put a camera on the tractor when cutting the grass, then collect images automatically of the entire field, do pixel-wise mapping of the grass and legume content in each image, then translate that to a biomass composition and get a legume content map of the field, which we then feed into the fertilization of the field. And the left-hand side is basically what I did my PhD in. So in order to validate all this work, we did 915 biomass samples. So that's a ½ meter by ½ meter, where we took a canopy image, so a camera looking straight down, and then cutting all the biomass, separating all the leaves to know the exact species composition of the dry matter. This we did over 3 seasons and 4 sites, so it's quite a varied set of data. It also includes the multiple grass species and multiple clover species, so it should represent at least a lot of forage mixed crops in Northern Europe. Looking at some data samples, we can see, well, 2 images here. It's pretty high resolution. It's about 5 pixels per millimeter, so you can actually see the tiny hair on the red clover. And it's pretty easy for us, at least, to see what's going on. So in the left-hand side, it's primarily grass, and the right-hand side, it's pretty rich in clover. And if we could take a look at the corresponding biomass label and see that we have approximately 72% clover of the dry matter in the right-hand side. Great. So looking at all this data, we have 915 biomass samples, and we have quite a varied validation set. Now, in order to train the computer to actually make use of this, we have multiple choices of detail. So we could train the computer to point out there's a clover in the image. That's the left-hand side. That's pretty easy to annotate, but we don't get that much value out of it because we don't really care about if there's a clover. We want to know how much clover do we have. The next sort of level in terms of annotation and detailedness is a bounding box detection. So we can basically count the number of clovers in the image. That gives us a little bit more value, but it doesn't exactly correspond to the canopy coverage of clover. So ideally, we want to know the exact number of pixels in the image, which is clover. That's pretty easy for the computer to learn, but it's pretty hard for us to annotate enough images for the computer to learn it. If we take a look at some image samples, so this is just a tiny crop of 5 biomass samples, and annotating this complex, heavily occluded entanglement of leaves is very time-consuming. It needs to be accurate for the computer to learn it accurately. So here we're talking about a couple of hours at least per image sample. So looking at the entire data set. So looking at the entire data set, we're talking about 10 years of work to annotate those. In Denmark, a PhD is not 10 years. So we had to come up with a different approach for that. And this is where the cleverness comes in, I would say. So instead of relying on thousands of manually annotated images, we want to do synthetic images instead. So we can have a soil background in the right here. Then we can do a small set of plant crop outs. So in top you see some clover leaves, you see some ryegrasses and a dandelion. So based on these crop outs, we can augment them, so rotate them, we can scale them, we can change the color slightly, and we can put them randomly into on top of the soil image, like this. Now, this itself doesn't give us that much value, but since we know what species it was beforehand, we can automatically get the exact pixel-wise label of that image as well. So with this approach, we can, based on a quickly made set of cropped images, we can create thousands of randomly populated synthetic images with accurate pixel labels. So first of all, that's very quick. So we can spend, let's say, 1 week doing a prototype data set and then get results. Secondly, it's accurate. So we don't have to rely on people doing a sloppy job annotating every pixel in the image. And we can also control the species distributions in the data set. So we don't get any biases, or at least we can control what biases we want in that data. Here we take a look at 2 synthetic images. So the left one is rich in legume, the right one is rich in grass, and we get the exact pixel-wise label of these two classes, like this. So once we have this, that's all we need to train a computer vision model, which we then do. So here we see two models. The left one is just for reference. That's an earlier model that I used. But the current model is the Deep Lab version 3+. Now, for people who don't know deep learning, it can be pretty complex, but it's basically millions of parameters where the network fits all these parameters based on the data in the image. So we don't tell the computer what a clover is. We just show the computer thousands of samples of clovers, and it will learn automatically how to determine if it's a clover or wheat or grass. So in short, let me just continue. For computer vision purposes, scale invariance is quite a big challenge as well. So when you look at the image to the left, you see a clover in the top right, down here, and you see a clover in the bottom part. Now these two clovers are pretty different size classes. For an agronomist, that's not a big problem. It's probably a white clover down here and a red clover up here. But for the computer vision, it's challenging to be invariant to all this. It also applies to different growth stages. It applies to different cameras or different camera heights. But this model has built in some sort of scale invariance using some computer vision tricks, basically. But that's probably too much detail to go into that. So if we go to some examples, so this is real images collected at some fields, and we can go look into what the actual performance of the model is. So here the grasses are marked with green, and the red is clover, and blue is background. And so based on the actual need at field scale, this is quite good, I would say. So this is definitely good enough for us to measure the legume content or to estimate the legume content, and then apply fertilizer based on that. We can go a little bit further. So we know that it works pretty well for distinguishing grasses and clovers. We can also do a little bit more and try to estimate the clover species. So in this case, purple represents red clover and yellow represents white clover. Now this is a much harder task. So you can basically teach any child to point out clovers and grasses, but it's pretty much harder to differentiate between red clover and white clover in images. And the model is sort of also falls under that challenge. But then again, there's just a proof of concept to show that we can do more than just this easy task of saying this is clover and this is a grass. Well, we cannot really rely on just for cherry-picked images. So we can take a look at all the biomass samples. We can process it directly through the computer vision model and pick out any, or determine every single pixel in every single image. That looks quite pretty. But if you take a look at the comparison between the actual clover fraction in the biomass versus what we found with the algorithms, we can see how well it generalizes across seasons and sites. So morphological filtering is or was the go-to approach before deep learning. So this was at the beginning of my PhD study. And now it doesn't work that great because it really depends on the size of the leaves. Since this is used from early in the season to late in the season, then the size of the leaves change, and we have to do a hand calibration per day. So that doesn't work when doing the entire data set. We can look at the first deep learning model that I made. This was pretty good advantage. It actually made it worthwhile to collect images and do this automatic segmentation. And then the deep lab model ended up with a really good performance prediction. So this is an R squared of 91%, which is definitely good enough for this to be deployed for actual fertilization maps. Great. Of course, since this is computer vision, it also relies on the quality of that image data that the model is fed. So this is just a few examples of people doing the same. So doing grass clover semantic segmentation with computer vision. And it was actually just to demonstrate and compare. So in the top right corner, it's drone imagery. It works quite well, but it's not perfect. In the top left, it's a robot from Australia, I think it is, or New Zealand. And the bottom right is a quad bike that we have in Denmark. So this quad bike, so if you remember, all the biomass images were based on handheld consumer cameras. We don't want to traverse all the fields manually on foot. So we built this special developed high-speed camera for agriculture surveying and put it on a quad bike. So it's a 5 megapixel global shutter camera with a powerful ring flash and a GPS and computer embedded. So basically you can drive up to 60 kilometers per hour and get images like this. So very good images, and you can collect them in a short amount of time. And you could have a look at what that looks like if it's working. And that's not working. Okay, so it's basically just quad bike is driving around 60 kilometers per hour, and it's automatically triggering the camera and putting the GPS coordinates into the image. So doing this on a field, that's quite easy. So every single point here is an image. The field is 11 hectare, and you just drive around back and forth, collecting images over the entire area. We could manually have a look at those images. So in the bottom left we see pretty much just grass. In the center image is just clover, and the right image is a mixture. So definitely there's some variation going on in this field. So we would benefit from mapping this variation. We could process these images with the computer. Here the grasses are blue and the clover is yellow, and that seems to work pretty nicely on these images as well. We can apply that on the entire set of images, and we see that the center part is quite rich in clover, and all the surroundings is a different picture. Again, this is only subsampling, so we need to do some interpolation to get some basis for our fertilization maps. And then based on current Danish guidelines on fertilization strategies, we can discretize this into to legume contents. So we can basically discretize into, is this below 20% legume? Is it 20 to 35% legume, or is it above 35% legume? Based on this, we can get a fertilization map to put directly into our slurry spreader. That's pretty much the end of this PhD project. So it turns out we can collect images, and we can process them, and we can do precision ag based on it. Now, we don't want to do just grass and clover. So we went to North Carolina to collect some images of cover crops. So this is more or less the same, but it's brassicas as well into the mixture. And as you see in the image, there's quite a lot of brassica here. It's an updated camera as well, but I won't go too much into detail with that. We can take a look at the images. So there's a lot of crop residue. There's some grasses, some brassicas, and some weeds down here as well. So I did the exact same approach, reused all the grasses, cut out, I think, 5 brassicas, a very small data set, just to try out if we can do the same for cover crops. And training the model and putting the images through, we get some pretty, I'd say, compelling results. So here the grasses are marked in red, brassicas in yellow, weeds in purple, and clover in brown. And the background is blue. So we get some pretty good estimates on the actual ground cover here. In the right-hand side, we see some really good results. Well, we do see some errors. For example, the grass here is marked as brassica, but overall it's good enough, I would say, to do rough mapping of the canopy composition. We can have some more examples here. So again, it generally works just based on 5 brassicas, and yeah, of course, it would work better if we had a more diverse data set. So that could include brassicas, which sort of has been partly destroyed, as these two in the left image. And it just include more weeds and more crops, basically, to improve the span of the synthetic data set. If we zoom out, we can do some similar maps as well. So here I try to map the plant coverage of these 2 cover crop fields in North Carolina. So we see that, first of all, the left-hand side field is more densely covered in live plant matter. The left-hand side, we have a big spot with more or less just bare soil or crop residue. Now, this is not that hard to do. We could do this with NDVI maps, but we can then go into the species composition maps as well. So if we look at the grass coverage, you can see that the right-hand side is actually more dense in grasses. And even more interestingly, if we go into the brassica coverage, we can also easily determine that the right-hand side field was probably not seeded that much in brassica. Maybe it was, maybe it wasn't, but at least we can see that there's a very big difference in the brassica part of these 2 fields. So we could go on and on about this. So we could go to a new task. We could want to map any sort of species, and then we would build a new synthetic data set and start all over again. We don't want to do that and just keep on every year. Instead, we are building this extensive repository. So this is in collaboration with Chris and Stephen. So we are seeding a lot of plants and tracking them and imaging them from emergence until about 1 meter or 1.5 meters tall. The first part of that growth is done in pots and then captured with a bench pot like structure. This happens in Texas, in North Carolina, and Maryland this year, and we're sort of starting to build this data set. So what we can do with this is that we can use this to build this extensive data set of both crops, of weeds, and hopefully also cover crop species. Something, well, to show you what this looks like is, well, here's an example to show what this looks like. So we're collecting images with a really high resolution Sony camera with the ring flash. But in order to organize all these images, we're combining them into an automosaic. So that is feeding all these images into an algorithm to build a bigger model, which sort of includes all this data, but at a lower resolution. So here you see some trial runs we're doing. So this is different plants growing in pots, and we're sort of testing out this equipment. Since it's an automosaic, we also get the height map as well. So this could be useful for determining the position of the plants or determining the position of pots in the scene. But going back into the color image, we can extract, for example, one plant here. Zoomed in, it looks like this. We can then find the corresponding high resolution image as a source for our synthetic imagery, like this. So you see that it's the same plant, but it's much higher resolution. You can see hair on the leaves, and you can see all these details. Then we can automatically or semi-automatically extract this plant, so we can reduce the background like this, and we can make a directly applicable plant model for our synthetic data generation. And this can be sort of augmented any way we want. And this is perfect for our sort of general purpose computer vision repository. We can do more than just semantic segmentation with this data. We can do weed counting. So this is a serial field. This is actually a Danish example, but we can do the same with our extensive weed repository. So we can basically overlay cropped out weeds into real images and then train a model to detect these weeds, not just if it's a weed or not, or also the weed species. We can also do, instead of counting the weeds, we can measure the sort of the weed infestation in terms of coverage. So this is a grass field where we have a lot of die-cut weeds as well, marked in red. So we get a pretty wide set of possibilities with this repository. But of course, since the computer is learning on its own, it will use all the features in high-resolution images to do the recognition part. That means that if we train a computer vision model on the left-hand side image, it will probably use the texture of the plants or of the leaves. It will use pretty much all the available information in that image. That's okay as long as we use high-resolution cameras. But if we, for example, use a GoPro camera, we cannot have a model trained on the Sony cameras in high resolution and then apply that on the lower quality images. So we also need to do some sort of controlled degradation of the image quality to sort of make it applicable for a wide range of cameras, if that makes sense. So that's a research question as well, a research topic that we are sort of going to address in this weed repository work. Because we don't just want it to work for the Sony camera, we want it to work for the GoPro camera, we want it to work for OD cameras, we want to make it work for drones as well. So we need to sort of control the actual quality of the images accordingly. Finally, sorry for taking this long, but finally, we're setting up a camera just for sort of info. We're setting up a Danish camera, if you say so, at Bach in Maryland. So we want to replicate this or extend this data collection with our technology into mapping cover crops and weeds in the US. And that's actually all I wanted to say to sort of encapsulate my work in PSA.